I really need like another hiccup. Oh, hey guys. So it's 9:35 on a Monday night, and I think it's a good time for us to talk about trial and death receptors, which relates to the cell receptor lab we did earlier this semester. So let's get to it. Cell receptors and their recognition molecules are the key players in a cell working with its environment and for cell-to-cell intercommunication. Right now, you're looking at a video of pathogen recognition in a macrophage cell. Receptors cover a wide array of functions within cells and require specific recognition molecules in order to be activated. Earlier this semester, we learned about lectins and how they recognize certain sugar residues on glycoproteins that run along the outer membrane of epithelial cells. What we will be specifically looking at today is trial, also known as TNF-inducing apoptosis ligand. Trial is a member of the tumor necrosis superfamily and is a type 2 transmembrane protein. It specifically binds to death receptors, which is the name given to receptors involved with tumor necrosis factors. When trial reacts with a death receptor, it forms a heterotrimer with 3 receptor molecules. Upon doing so, it causes conformational changes in the receptor molecule that allow adapter proteins to bind to them. These adapter proteins enable procaspase 8 to bind, which then becomes caspase 8, beginning a long downstream signaling cascade for apoptosis to occur. Now I realize the video behind me is very attention-grabbing, but I will need you to stay with me for just a few more minutes. Trial binds to 5 different receptors, of which only 2 are capable of inducing apoptosis: death receptor 4 and death receptor 5. The remainder of the receptors are decoy receptors, so they basically lack effector domains and cannot induce apoptosis. It is currently thought that they are necessary to mediate and inhibit trial's function, so that way it doesn't happen out of control. Trial's significance as a signaling molecule lies in the fact that it appears to selectively activate apoptosis in cancer cells, although more research is being done into this. Currently, there is much research being done to try and utilize trial in some way for anti-cancer treatment. One such study is the one this project was inspired by. At the University of Groningen, researchers were trying to create trial mutants that were selective for the functional death receptors. Previously, trial-specific mutants for death receptors 5 have been made, but until now, functional ones for 4 haven't. The researchers created a set of 21 single mutations that were modeled to show the increased affinity for death receptor 4 they were looking for. These mutations were also combined with certain mutations for death receptor 5 to see if increased affinity for both receptors could be maximized. The researchers utilized techniques such as live microscopy, immunoprecipitation, and modeling and computational protein design using the program FoldX. The result of their experiments were very pleasing. A remarkable capacity to induce apoptosis was seen in several cancer cells in vitro, including a panel of human colon adenocarcinoma, Burkitt's lymphoma, pancreatic carcinoma, human ovarian carcinoma, and human breast adenocarcinoma cancer cell lines. The variants were also tested on human normal fibroblasts, and no damage was shown to be done. This further strengthened their idea that the increased apoptotic activity on cancer cells had no effect on normal cells.
For more information on this study, you can search trial on the scientific journals Science, Nature, and Cell. That was our presentation. Thanks for watching, and good luck to everybody else on their presentations.